As we all know, DNA is pivotal to the growth, reproduction, and health of all living organisms. It contains the instructions necessary for cells to produce proteins that affect many different processes and functions in living organisms. Good day everyone, my name is Paula Aliposa, and today we will be performing an easy experiment of genomic DNA extraction of plant cells using readily available household items. For this experiment, you will need the following items: a blender, cold 70% ethanol, detergent, distilled water, droppers, pineapple juice, plant leaves, plastic containers, strainer, table salt, and a weighing scale. The first step is to prepare our plant samples. For this experiment, we used the Pomea aquatica leaves, commonly known as Kangkong, which is the most abundant lowland vegetable in the Philippines. First off, we wash the plant leaves by running it under tap water. We then weigh 50 to 100 grams of the leaves and transfer it to a blender and blend it with about 100 ml of ice-cold water and 2 teaspoons of table salt. Plant cells are surrounded by a wall made up of a super strong molecule called cellulose. Because of this, we apply physical force on it by blending the leaves, which breaks apart the cell walls of the plant. Table salt or sodium chloride removes proteins from the cell and the DNA and keeps these proteins dissolved in the aqueous layer so they don't precipitate in the alcohol along with the DNA, which we will be adding in the latter parts of this experiment. Sodium ions also create a temporary attraction between the positive sodium ions and the negative phosphate backbone of the DNA, neutralizing the DNA and making it more hydrophobic, which ultimately allows it to be easily dissociated with water in latter steps as well. Second, we filter the solution into a new container using a strainer. In this step, we separate the plant DNA from cell debris and other insoluble materials. After filtering out the debris, we add 2 tablespoons of liquid detergent or dishwashing liquid to the solution. We then stir it gently and allow it to stand for 5 to 10 minutes. We know that the DNA is contained within the cell's nucleus, which is surrounded by the cell membrane. We also know that the cell membrane is made up of phospholipids, which are similar in structure to fats. In this step, detergent molecules dissolve the cell membrane of the plant cells by interacting with the lipid particles in it, which ultimately exposes the DNA. Using a dropper, we transfer a smaller amount of the solution into another container. For every 2 pinches of the solution, we add 1 pinch of ice-cold pineapple juice. We then mix the liquids together by gently agitating the container for another 10 minutes. Basically, pineapple contains bromelain, which is a protease enzyme that breaks down histone proteins bound to the plant's DNA. And based on experience, it is best to use fresh pineapple juice rather than canned or bottled pineapple juices, for they may have already been processed, causing these essential enzymes to be removed or diminished. We then gently add ice-cold 70% ethyl alcohol to the solution. The amount of ethanol to be placed in the solution must be approximately the same as the amount of pineapple juice added. With the addition of alcohol, DNA clumps together to form visible white gelatinous clumps of precipitate, and this happens because alcohols are non-polar solvents. So, DNA becomes more hydrophobic and the sodium-DNA bond becomes even stronger, which eventually causes DNA to precipitate out of the aqueous layer and onto the alcohol layers. In simpler terms, DNA becomes insoluble in the solution if the said solution contains both alcohol and salts. Now in this step, it is important to use cold ethanol because warm alcohol causes DNA to denature or break down. And if this happens, our entire experiment would be regarded as useless. Finally, we then separate the DNA from the rest of the solution by spooling it into a wooden stick.